Hello chemistry fans, and welcome back to chemistry problem solving session here at the Academic Success Center on Broward College North Campus. I am your host, Dr. Russell Betts, and I will be your guide through this problem today. So I see we're dealing with proteins. In protein structure, what is meant by the following terms? Primary structure, secondary structure, tertiary structure, and quaternary structure. All right. Now, protein is just a series of amino acids. Usually, generally considered more than 50 amino acids is where the proteins begin and peptides stop. So let's see here, primary structure. Now, primary structure is basically the sequence of amino acids in a protein. What comes after what? So what amino acid follows what amino acid? So for example, glycine, alanine, threonine, serine could be a primary structure, just the sequence of amino acids. So it's the sequence. That's it. Sequence of amino acids, if you want to say, in a protein. Or in a peptide. It doesn't have to just be in a protein. It could also be in a peptide. So that's the primary sequence. Just what amino acid follows what in the series, or the sequence of the amino acids. Secondary structure. Secondary structure are the things such as alpha helixes, beta pleated sheets, and triple helix. Oops, H-E-L-I-X, there we go. Triple helix. These are different types of structures that are formed as a result of hydrogen bonding of the amides of the amino acid sequence, or the amino acid backbone. So they're the hydrogen bonding of the amides of the protein. In other words, it's not the side chains. It's the amides that are hydrogen bonding to one another that are causing these three types of secondary structure. Now, an alpha helix, if you want to think about it, is like a telephone cord, the old-fashioned telephone cord that would coil up and you could stretch it out and it would coil right back. That's the type of alpha helix. A beta pleated sheet, imagine if you just put numerous pleats or numerous folds into your sheet. That would be a pleated sheet. Or a triple helix is just three helices basically interwoven onto each other. And those are as a result of the hydrogen bonding of the amides. It's the amides that are doing that, not the R groups of the amino acids. It's the amides that are doing it. Tertiary structure. Now, tertiary structure is the folding that a protein does in order to stabilize itself, and that is caused by the R group interaction. Now, there's different ways the R groups will interact, which we'll go over in a minute. But for now, tertiary structure is the folding of the protein into a stabilized compact structure. Now this is caused by R group interactions. Now, the secondary structure is not the R groups. The secondary structure is the amides hydrogen bonding. The tertiary structure is caused by the R groups, not the amides. Okay? That's the difference. This is caused by the amides interacting, hydrogen bonding for the most part. tertiary structure is caused by the R groups interacting. And we'll go over in a minute, in a subsequent video I should say, what those interactions are. Now a quaternary structure, that occurs when two or more polypeptides, or two or more small proteins, come together to make a bioactive protein. When I say polypeptides, you can think of it as small proteins. Polypeptides combine to make a bioactive protein. And that's a quaternary structure. You can bet money that the interaction between the polypeptides is hydrogen bonding and all the other kinds of hydrophobic and hydrophilic interactions. But it's two separate polypeptides that have found each other and have come together to make a bioactive compound. All right, now I hope this helps clear up or help you, or will at least help you to remember what the four types of protein structure are. Please make sure you read this in your book as well. Don't just watch the videos. It's important for you to read as well to make this stuff basically more real to you. It's important that you understand these things. It's a very important concept in biochemistry on how proteins fold and how they work. So please familiarize yourself with it and be ready for it on an exam. All right, now with that, I'd like to wish you good luck and good chemistry.